And so good evening and welcome to the latest installment of the SLAC Public Lectures. So today we're very happy to have Peter Dahlberg, who's doing some, really beautiful stuff that I'm sure you'll be eager to learn about. Peter got his bachelor's degree from McGill and then came to the University of Chicago and got into the biophysics lab there, where he did some quite interesting imaging projects. And then after that, he came out here to Stanford in 2016 to work with actually two of our imaging giants here at Stanford. Wah Chiu, who is a big expert in what's called cryo-electron microscopy, and W.E. Moerner, who's actually a Nobel Prize winner for what's called super resolution spectroscopy. And the question is whether you could actually bring these things together and learn more and more about the inner workings of cells. So that's what Peter will talk to you about today. And one more thing. In 2021, Peter was awarded our Panofsky Fellowship, which is very competitive, but let's just say a very nice deal for those who win it. And so now, let me turn it over to Peter and he'll tell you all about what's inside the cell.
All right, well, thank you very much for that kind introduction. It's my pleasure to be here today to talk to you all about some of the work that I'm conducting up here at SLAC National Accelerator Laboratory. So oftentimes our job as scientists is to observe the world around us to try and understand how things work. And for me, I'm very interested in understanding how cells work. But I can't go out and observe cells. I need tools. I need microscopes to magnify these cells so that I can observe them, try and understand how they function. Unfortunately, there's a fundamental limit to the resolution that I can obtain with a traditional light microscope. And that limit prevents me from seeing much of the inner workings of these cells that I care about. So today I'm going to be talking to you about two advanced microscopy methods that, when combined, can give a very clear picture of the inner workings of cells. Now, before I get into the details of these advanced microscopy methods, I want to begin with a brief introduction, and this is largely for the students in the audience, to remind them that there is no stereotypical scientist. We come from all sorts of different backgrounds. So I myself was born and raised in Minnesota. I loved being outside. I loved fishing. Here I am out on one of our 10,000 lakes. And like many kids, I had an early interest in science. And also like many kids, early on, this manifested as a love for dinosaurs. So here I am, about 3 years old, obviously dressed up for Halloween as a dinosaur. I grew up. I went to high school. Here I am at homecoming in 2006. This is actually my future wife, so you'll see her again in a few pictures. And from high school, I went to McGill University, as Michael said, to study physics. So I spent a lot of time in the Rutherford Physics Building, working late nights on problem sets. I know that building far too well at this point. And while I was at McGill, I had a very fortunate opportunity, actually provided by the Department of Energy, to do a summer research internship in an area of science known as biophysics. So this is a branch of science that uses physics tools and quantitative approaches to solving problems to attack biological questions. And I realized this was a very transformative experience for me because I realized that this was an area of science that I really loved. So I left McGill University, completing my degree, and went to the University of Chicago to work under Professor Greg Engel here. And in Greg's lab, I got to use laser systems like this one shown here. This is an ultra-fast laser, which means it produces very short pulses of light on the order of a millionth of a billionth of a second in duration. And we use these very short pulses of light to act like a fast flash on a camera, to follow very fast dynamics. And the dynamics that we were concerned about were related to how photosynthetic organisms absorbed and harvested solar energy. Completing my Ph.D. with Greg, I like to joke that I moved from ultra fast to ultra small, because as Michael said, I left the University of Chicago to come to Stanford University in late 2016, and this is really where my microscopy journey begins. And as he said, I joined Stanford as a postdoc to work at the interface of two different research groups, that of W.E. Moerner and Wah Chiu. And these two gentlemen are really experts in their respective microscopy methods. So W.E. Moerner is an expert and early developer of super resolution fluorescence microscopy. And as Michael said, he got that Nobel Prize in 2014 in chemistry for his foundational work in this area. And Wah Chiu is a real expert and pioneer and developer of cryogenic electron microscopy. So these are the two methods that I'm going to be bringing together later on in the talk: cryogenic electron microscopy and super resolution fluorescence microscopy. And I've enjoyed my time so much at the interface of these two groups that in some sense I've just refused to leave. So about a year ago, as Michael said, I joined SLAC officially as a Panofsky Fellow to continue the work that I started at the interface of these two research groups. And SLAC has been a wonderful place to work. So despite having my hands very full at home right now at the moment, when I come into lab, when I find the time to come into lab, I get to work with some of the most advanced microscopes that the world has to offer. And I can see and do things that no one's ever done before. But before I get into the details of what we're able to do in the lab and how we're doing it, I want to begin with some foundational knowledge. And because this is a microscopy lecture, I want to make sure that we're all on the same page about small units of length. So on the next slide here, I'm going to go through some units of length with some objects of reference. And so the first unit I think we're all familiar with, it's a meter. And as a good object of reference, we have Steph Curry. So Steph Curry is 1.88 meters tall, according to Wikipedia, and is unfortunately far larger than the systems that we're going to be looking at today. So the next unit that I'll introduce is the millimeter. So this is 1,000th of a meter. And a good object of reference is your fingernail. So if you look at your fingernail is actually about 0.5 millimeter thick. But the systems we look at are far smaller than this. So I'm going to introduce the next unit, which is a micron. So a micron is 1,000,000th of a meter. And here it becomes challenging to find objects that we interact with on an everyday basis, because a micron is actually too small for you to see with your naked eye. But a good object of reference might be the width of a fine human hair. That's about 50 microns in width. But the systems and the length scales we're concerned about are far smaller than this. And we have to go down a few more orders of magnitude and introduce the nanometer. So a nanometer is 1,000,000th of a meter. And now we're far beyond what we could see with our eyes. So there really is no everyday object that you might interact with that's on this length scale that you can see and sort of visualize. So perhaps a good way to understand just how small a nanometer is with the following ratio. So the width of a human hair to the height of Steph Curry is roughly the same as a nanometer is to the width of that human hair. So you can imagine for a moment sort of stacking up fine human hair to reach the height of Steph Curry. The number of hairs that would take is roughly the same number of nanometers that it would take to span that human hair. So this is an incredibly small unit of length, but it's going to be relevant for the systems that we're looking at today. So with that foundation in place, I now just want to watch a few fun videos of cells doing the incredible things that they do. And all these videos that I'm about to show you are taken with what I would call traditional light microscopy methods. So the first video I want to show you is of a white blood cell, shown here in the center, moving through a sea of red blood cells, these structures here, and it's chasing this bacterial cell. And note that the scale bar here, this is roughly half the width of a human hair. So here, I'll play that video. And what we can see is that it really looks like this white blood cell is chasing that bacterial cell. It's following it through the sea of red blood cells. And what it's doing is it's following a chemical gradient left behind by that bacterial cell. And its job, what it's trying to do, is sort of trap and engulf that bacterial cell in a process called phagocytosis, where it's going to engulf it right there, and then it's going to begin digesting that bacterial cell. This is one way that your immune system works to keep you free of foreign cells. This next video, what we're looking at are parasite cells. So there's a few parasites here. There's one here, here, and two over here. And this type of parasite is called Toxoplasma Gondii. So Toxoplasma is related to the parasite that causes malaria, but it leads to much fewer health complications. It's much less hazardous. But in a lot of ways, it's a much better parasite. So Toxoplasma Gondii is known to infect about 2 to 4 billion people worldwide. So there's a good chance that members in the audience today have this parasite inside their cells. If you have cats, you're much more likely to have this parasite because one of the processes in this life cycle is spread through cat feces. But what we're going to be watching in this video is one of these parasite cells invading a host cell. So right here, this sort of textured surface is a human cell. And what we're going to see is this parasite here, keep your eye on that one, is going to glide along the surface of this cell and then pull itself in as it invades that host cell. So here it's sort of looking around, it's attaching to the surface, it's gliding, and then right there, you can actually see it's pulling itself in to that host cell. So watch right here, there's sort of a junction as it squeezes itself in. So what's not shown in this video is after this cell invades this human cell, it's going to begin replicating itself, and it's going to produce several copies of itself. And then these cells are going to burst out of that human cell and go off and infect other cells. So these are sort of fairly complicated cells, Toxoplasma Gondii and that white blood cell. But even if we look at sort of the simplest cell, a bacterial cell, we still see incredible things. So here we're looking at a bacterial sample. Note the scale bar at the top. And there's several bacteria, or many bacteria, I should say, in the field of view here. And most of them are sort of fixed and not moving. But when I play this video, you'll see others zipping around in this cell culture. And so much like a dog maybe finding its way into a kitchen while you're cooking, these cells are following chemical gradients. They're sniffing around for food in regions where they want to grow. So watching these videos sort of immediately makes me think of many questions, right? How do these cells sense their environment? They don't have a nose or ears or eyes like you or I, and yet they can sense the environment around them, right? And how do they decide what to do if they have all that information? How can they make a rational choice based on their environment? They don't have a brain. And yet we looked at that white blood cell, it was clearly chasing that bacterial cell. And how do they move, for that matter, right? They don't have a skeleton, they don't have muscles. How are these cells moving? So answers to questions like these and these questions eluded scientists for a long time. And the reason that answers to these questions eluded scientists is that the answers to these questions really rely on structures that are far smaller than the cells themselves to understand. So here I have a cartoon picture of a bacterial cell. It's only about 2 microns in length. And in this cartoon depiction, we've colored in many of the different structures that we might see, things like a cell wall and different proteins shown in purple and blue. We have DNA and RNA shown in yellow. If I zoom in one more time, we can see these things more clearly. And these biomolecules, as I'll call them, all these different components, especially these proteins, these blue and purple components, are responsible for carrying out these complicated functions through their complex interaction of these different biomolecules. But as I said at the top of the talk, there's an unfortunate resolution limit in what we can see with traditional light microscopy, and that obscures much of what's going on at this fine length scale. And that resolution limit is what we call the diffraction limit. So let's try and understand that a little bit better. So the diffraction limit can be defined as when 2 objects get closer and closer together in my microscope, eventually they become indistinguishable. And that separation at which they become indistinguishable is what I'll call the diffraction limit, where I no longer can resolve these 2 separate objects. And an understanding of this was first worked out by Ernst Abbe in 1875, actually working with optics made by Carl Zeiss. So those of you who have worked with optics before might recognize that name, Carl Zeiss. Zeiss is still a big company in terms of optics. But what Ernst Abbe figured out in 1875 is that diffraction limited resolution, that separation, was related to the wavelength of light that we were using, this lambda, and it's roughly half that wavelength of light that we're using to image our sample. And no amount of improved optics, better lenses, better objectives will get past that limit. That's a fundamental limit with the way that we're doing our imaging. So we're familiar with light being a wave, and that wavelength of light for the visible spectrum is between 400 and 700 nanometers. And that leads to that diffraction limit of being roughly 250 nanometers. So if we come back to that cartoon depiction of the bacterial cell, we don't see this beautiful cartoon image when we look in our microscopes. We see something more like this. And that's because of that diffraction limit. So if we want to understand how these different biomolecules are interacting and working, we really need to employ some new microscopy methods that go beyond this resolution limit of 250 or so nanometers. So that's really the motivation for our work in cryogenic electron microscopy and super resolution fluorescence microscopy, which I'll talk more about in just a second, beginning with cryogenic electron microscopy. So you might imagine that one way around this limit of 250 nanometers is just use light of shorter and shorter wavelengths to do your microscopy, right? And people have built X-ray microscopes, that's the thing. But another way to do this is to use electrons rather than photons to do your microscopy. So quantum mechanics tells us that we can think of electrons as a particle, as we oftentimes do, but we can also think about them as waves. And it turns out that the wavelength of an electron is incredibly short. It's related to the inverse of the momentum of that particle. So the faster that my electrons are moving, the more momentum they have, the shorter the wavelength is of those electrons. And in our electron microscopes, the electrons are going to be moving very fast, about 70% the speed of light. And that leads to a wavelength of just 0.001 0.002 nanometers. So in terms of our electron microscopy, we're no longer going to be limited by the diffraction limit if we want to resolve these biomolecular structures. So how do we build an electron microscope? Well, in many ways, the working principles of an electron microscope aren't all that different than a light microscope. So here's a diagram of an electron microscope, and many of these components that have been labeled have analogous components in a light microscope. So you might be wondering, well, the first question is, where do I get my source of electrons? And that also isn't all that different than how we get photons in a light microscope. There's several ways to do it, but one of the earlier ways to do this was just with a hot filament, so something that would look like this. And you'd heat up that filament to very high temperatures, and the electrons would get enough energy that they would fly off. And at this point, they're actually moving fairly slowly. And so the first thing that we do is we accelerate electrons like that with a high voltage, several hundred thousands of volts. And so now our electrons are moving very fast at 70% the speed of light. And now we need to focus them and manipulate them to actually image our sample. And we can't use something like a glass lens like we would in a light microscope. The electrons actually won't go through that. So instead, what we do is we use electromagnets to focus and manipulate our beam of electrons. And the reason we can do this is something called the Lorentz force, which comes from physics, which tells us that charged particles moving through a magnetic field feel a force. And we're going to use that force to focus and manipulate our beam of electrons. So we take our electrons and we are focusing them and moving them down through the column of this microscope. We pass them through our sample, and we're going to image them on a detector. And even that detector, its working principles aren't all that different than the CMOS detector that you might have in your camera phone. Now, there are some pretty big differences when we're working with electrons rather than photons. And one of the big ones is that electrons interact with matter quite strongly in comparison to photons. And so this leads to a few things. Electrons have to be, or sorry, the sample has to be quite thin in order for our electron beam to penetrate through the sample. And also this entire microscope column has to be in vacuum, because the electrons would scatter off of the air if there was air inside that microscope column. And that leads to a big problem, right? Because last I checked, you, me, all of us in the audience would much rather be in atmospheric pressure than in vacuum. So how do we study biological systems if we have to be in a vacuum environment? Well, one way that we've figured out how to deal with this is that we freeze the sample first and then load it into the microscope. And that's what I'll call cryogenic sample preparation. So the way that we prepare samples at cryogenic temperatures is the following. We begin with an electron microscopy grid. So an electron microscopy grid is this metallic mesh that I'm showing you here. It's about 3 millimeters in diameter. I actually have a few. So after the lecture, I think out in the hallway, you guys can take a look and try and play with and handle these electron microscopy grids. They're quite difficult to handle. But it's a metallic mesh that's about 3 millimeters in diameter, and stretched over the top of that metallic mesh is a very thin layer of amorphous carbon. And that sounds complicated, but that amorphous carbon, really think of it almost like graphite or pencil lead, a very thin layer of that. This is what it looks like in cartoon form. In a light microscope, these EM grids or electron microscopy grids look something like this. And this is a different type of image, but it's showing you the surface of that electron microscopy grid and the surface of that amorphous carbon film. And what you can see is it's not continuous. It has many small holes cut into it, and those holes are on the order of a micron in diameter. And our best electron microscopy is really going to be done in these holes where there's no carbon layer to obscure our transmitted electron beam. So to prepare a sample on these electron microscopy grids, what we do is the following. We take cells, deposit them on this electron microscopy grid, and our cells are typically in some solution. And so we first have to remove the majority of that liquid, and we do that by touching a piece of filter paper to the electron microscopy grid, and that wicks away most of the liquid, leaving behind this very thin sample that's hydrated. We then take that thin sample and we plunge it in the cryogenic liquid ethane or propane. And that liquid is kept at about minus 180 degrees Celsius or minus 290 degrees Fahrenheit. And this very rapidly fixes and freezes the sample and preserves it in its native hydrated state. So this is what it looks like in cartoon form. In practice, it looks something more like this. So here you're looking at a pair of tweezers that are holding one of these EM grids, electron microscopy grids. And on this grid is a small droplet of bacterial cell culture. And here are the two pieces of filter paper that are going to come in contact with that grid and wick away most of the solution, leaving behind a thin layer of cells. So here's a fun slow motion video of what this process looks like. So here those pieces of filter paper are coming together around that grid, pulling out most of the liquid, leaving behind that very thin layer of cells. And then in a second, those two pieces of filter paper are going to pull apart, and the grid and the tweezers are going to plunge down the hole into a pot of liquid ethylene. And so now that sample has been frozen. So this is how we prepare samples for an electron microscope. So the next step in this obviously is going to involve an electron microscope. And this is where SLAC has made a huge investment in recent years. So SLAC has invested heavily in the CryoEM Center, largely driven by the Wah Chiu. And so in recent years, we've acquired 7 state-of-the-art electron microscopes, and this has led to the development of 2 National Institutes of Health centers here. We have sample preparation facilities that allow us to prepare samples by that method that I just described, plunge freezing, as well as a few other methods of freezing. And we have lots of expert staff to help the place run smoothly. This has led to dozens of users from Stanford and many more from around the country and really around the world coming to SLAC to perform cryogenic electron microscopy. It's also led to numerous workshops and trainings that have impacted hundreds of researchers around the world. So what am I doing with these electron microscopes? I'm doing a type of imaging called cryogenic electron tomography. So the way that cryogenic electron tomography works is the following. What I'm doing is I'm going to take my cellular sample, I'm going to put it into the electron microscope, and I'm going to take a series of projection images with the sample held at different angles relative to that transmission electron beam. And so from this series of projection images that I take, I can work backwards to calculate a 3-dimensional volume. And the math here that we're using isn't all that different from what's done to generate an MRI image. So this is sort of the cartoon depiction of that. Again, in practice, it looks something like this. So here we're looking at a bacterial cell. This bacterium happens to be a Caulobacter crescentus cell, an organism that we'll talk more about in a few slides. But this bacterial cell is going a process of division. So we have a daughter cell over here that's just about to split from this other daughter cell, which is over here. You can see the hole, and so this is all frozen on one of these electron microscopy grids. You can see the hole here, which is that hole in that amorphous carbon film. So right here is just ice. You also see in this sample a bunch of these small black specks. And these are small gold beads that I add to the sample that allow me to align one projection image to the next. So as I play this video, what I've done is I've just stitched all those projection images into a single video, and that looks something like this. So it's from this tilt series, as I'll call it, that we're able to reconstruct a 3-dimensional volume. So what that looks like for these bacteria is something like this. So I apologize, this is a different cell. It's the same species, but a different cell that you're looking at now. But this is a reconstruction of one of these Caulobacter crescentus cells. And so again, there's a lot going on outside the bacterium, but if you just draw your attention to the periphery of the cell and the structures that you can see on the interior. So this is just a single slice from a 3D reconstruction. So as I play this video, what we're going to do is we're going to go sort of slice by slice and look at how these different structures are arranged within the cell. And that looks something like this. In any of these different structures that we can identify, we can color in. So things like ribosomes are shown in yellow. These are proteins, or these are structures rather, that are responsible for converting RNA into proteins. We have a surface layer of proteins shown here in green. This is sort of like the shell of the bacterium. We have 2 membranes that we can see, the inner and outer membrane, shown in maroon and cyan. We also have these orange spherical structures, these granules. These are sort of like a carbon or phosphate storage for the cell, or kind of like a sugar storage granule. We also have these structures down here. These are pili, these protrusions here. They can sense surfaces around these bacteria. We have a flagellum here, this structure, which is sort of a whip-like structure that spins around very rapidly and propels the cell as it's swimming in the solution. And we have this structure here, the small red structure, is known as the chemoreceptor array. It's a set of proteins that can actually sense the external environment around this cell and provide feedback to this flagellum to sort of direct where the cell is going. So all of this incredible subcellular resolution is made even more impressive when we compare this to what we would see if we looked at the same cell type in a bright field microscope. So this is imaging a Caulobacter crescentus cell just in bright field. So clearly, the researchers of a few hundred years ago or 100 years ago really would have had a hard time understanding all the processes that were going on within the cell. So as another example of what we can do with cryogenic electron tomography, I want to show some images from Toxoplasma Gondii. So here is actually a still image taken out of that video that I showed you before. So this is the parasite cell here. And much of the machinery that this parasite uses to invade host cells or human cells is housed in what's known as the apical complex. It's the very tip of this parasite here. And that apical complex is incredibly small. It's only about 500 nanometers across and about 500 nanometers long. So it's essentially a diffraction limited spot. No matter what we do in traditional light microscopy, we're not going to get an image much better than this. But now if we take these parasites and we image just that apical region of these cells, we can see something more like this in electron tomography. So again, this is a single slice from a tomographic reconstruction of these parasites. And the way that we get these parasites is we've ripped them out of the human cells that they were living in. So in this slice, you can see a lot of cell debris out here. This is sort of cell debris from the human cell that this parasite was in. But then the parasite itself is sort of this structure here. So this right here is the apical complex of Toxoplasma Gondii. So again, as I play this video, we're going to go slice by slice and see how these different structures are arranged in three-dimensional space. So in the case of Toxo, we can see different structures than we saw, of course, in that Colobacter cell. So we can see things like microtubules. That's these structures that are being colored in blue. These act sort of like the skeleton of the cell. We can also see these other structures labeled in green called micronemes. We can see these rhoptery shown in red. We can see these apical vesicles, as I'll call them, in yellow. And these different green, well, all these structures really, but the green and the red and the yellow structures really play key roles in invading these host cells. We're not sure all of the details of this process and exactly how this organism performs its invasion process, but we know that these structures are important. And one of the focuses of this paper in 2022 was looking at an interesting interaction between these rhopteries, these red structures, and these apical vesicle structures shown in yellow. And what we found in that work is that if we take parasites and they're resting, meaning they're not about to invade any host cells, there's a certain arrangement to these rhopteries and apical vesicles. It looks something like this. So again, looking at these red and yellow structures, a zoom in is shown here. What we can see is that these rhopteries and apical vesicles are in close proximity, but it doesn't actually look like they're interacting or touching. But if we take these parasites and we chemically stimulate them to tell them that they're about to invade host cells, that they're sort of primed for invasion, then we see a different interaction between these structures. So we see our rhoptery coming down here in red, and then we see the apical vesicle here. And in that zoom in, you can see that there's a very clear pinching of this apical vesicle and an interaction now between that apical vesicle and the rhoptery. And this interaction had never been seen before. And you might look at this and imagine that this might be a good target for therapeutics, because we know that this interaction is sort of part of the process in host cell invasion when we are seeing this interaction only when we prime these cells for invasion. So that's the type of information that we can get from electron tomography. But if I think about that question of using this information for therapeutics a little bit longer, take a step back and look more critically at the data, well, there's a challenge. Because what I can see is I can see these different subcellular structures, right? I see these rhopteries, I see these vesicles, I know they're interacting, but how would I target that, right? How would I target this apical vesicle and try and disrupt this interaction to try and make some therapeutic? One of the challenges I have in doing this is I don't actually know which biomolecules, which proteins are associated with that apical vesicle or with that rhoptery. At this point, you might be saying, but I thought you said you had 0.002 nanometer wavelength, that you could resolve all the biomolecules. Well, it turns out electron tomography isn't limited by diffraction of the electrons. It's limited by something else. The resolution of electron tomography is limited by something that we call radiation damage. So as these very energetic electrons come in and interact with the sample, they do things like break bonds and degrade that high resolution information very quickly. And in practice, that limits the resolution of electron tomography to more like 4-ish nanometers. And that resolution is too low to identify many of the biomolecules of interest. So to understand this a little bit better, I'm going to turn to this image of Colobacter crescentus, this bacterial cell we were looking at before. And what I'm going to do is I'm going to zoom in on this end of the bacterium here. So we can see the things that I mentioned before. We can see the ribosomes that I talked about that convert RNA into protein. We can see the surface layer proteins, but we also see this black void where it looks like there's nothing inside the cell. But we know that there's something there, right? In some sense, if I look at the cartoon picture, it's in some ways more accurate, right? We can see all these different proteins, for example, in the region where we expect to see this void in electron tomography. So those proteins are there, those different molecular machines are there, but we can't identify them from electron tomography alone. So to impress this point just a little bit further, so Caulobacter expresses about 1,000 different proteins. And we can see the different structures that we can identify and label here. It's only about 10 of them. So the majority of these biomolecules aren't identifiable from tomography. And this really motivates turning to our next method, which is fluorescence microscopy. So what is fluorescence and how can we use it for microscopy? So fluorescence is a property of, say, dye molecules, like the chemical structures I'm showing here, absorbing light of one color and emitting light that's redder. So these different dye molecules, which are all on the order of about a nanometer, these are their sort of various chemical structures, dissolved in solution down here, excited with UV light. These are the different colors that they emit, everything from blue light all the way out to red light. This is the fluorescent spectrum of these different dyes. So that's fluorescence. We can excite with one color and we get out a different color. How do we use that in microscopy? Well, we've come up with many clever ways of attaching fluorescent molecules to specific biomolecules of interest. So here, imagine for a second that we want to know where this square protein is in our cells. What we can do with fluorescence microscopy is we can take one of these fluorescent labels and we can specifically attach that to all the square molecules in our cell. So now whenever we see fluorescent light of a certain color, we know that one of these square proteins of interest is in that region. Now, this alone doesn't give us anything beyond the diffraction limit, but it does tell us where specific biomolecules are in our cells. And so, as I said, it doesn't pass the diffraction limit. So if we take these proteins and we put them in front of our microscope, what we see is a diffraction limited spot that's several hundred nanometers across, even though that single dye is only a few nanometers across. That's due to the diffraction limit. But we can do some pretty powerful things with diffraction limited fluorescence microscopy. As an example of this, we can watch some human cells divide. So here, what we've done is labeled microtubules in blue and DNA essentially in yellow. And what we're watching in this video is the DNA get partitioned into the two daughter cells. So that looks something like this. You can see sort of equal copies of the DNA getting pulled into the two daughter cells. But what if we want to go beyond the diffraction limit with fluorescence microscopy? Then we have to turn to something called super resolution fluorescence microscopy. And there's several variants, several different types of super resolution fluorescence. The one I'll talk to you about today is what I would call single molecule-based super resolution. And all single molecule-based super resolution approaches really rely on two key concepts. The first key concept is this idea of single molecule localization. So if I have a single bright emitter in an otherwise dark field of view, what I measure on my camera is some broad intensity profile like this one in the pixelated image I showed on the previous slide. But I can determine the position of that individual emitter much more precisely than the width of this intensity distribution. The way that I do that is I take that intensity profile and I fit it to a model function. And now, in some sense, what I'm trying to do is I'm trying to find the peak of this mountain here. And I can do that with a precision that scales as 1 over the square root of the number of photons that I collect. So the more photons I collect, the more precisely I can determine the peak of this mountain, which is sort of an indicator of the position of that individual emitter. Now, this first key concept in and of itself doesn't give me a super resolved image. All it does is tell me very precisely where an individual molecule is. So to take a super resolved image, I have to add in the second key concept, which is this idea of active control. So imagine for a minute, I have some extended structure like this helix shown here, and it's densely decorated with fluorescent reporter molecules, these gray circles. And if they're all emitting at the same time, then what I see is just this diffraction limited haze. And I can't play this trick of the first key concept of single molecule localization. But if I had some ability to turn off the majority of molecules in the sample and leave on only a sparse subset, then I could localize that sparse subset of molecules. And if I further had the ability to turn off those molecules and turn on the next random subset of molecules, then I could localize that next set of emitters. And through time, I could build up a pointillistic reconstruction, which is my super resolved image. Now, people have come up with many clever ways of controlling the emissive state of molecules. But what I want to show you next is just an example of what this super resolution data looks like. And I'll apply it to these microtubules that we were just looking at. So here in the center is a diffraction limited image of microtubules that have been fluorescently labeled, roughly corresponds to a field of view like what's shown over here in the red box. In this cell, we're going to take a video of it. And this cell won't be moving around. We've chemically fixed it and killed it. So there are no more dynamics of the cell moving around. What we're trying to do is take a really clean, high-resolution picture of these microtubules. And so in that video of these microtubules, an individual frame looks something like this. So here what you're seeing is that sparse subset of molecules that are emitting. And each one of these bright spots is a single molecule that we can localize. And I'm going to put that localization over here on the right. The next frame of the video might look something more like this, where I have another set of localizations that I've plotted over on the right. In the next frame, another set of molecules. In the next frame, another set of molecules. We take many frames like this, and through time, we build up that pointillistic reconstruction, which is our super resolved image on the right. And what you can see if we compare these boxed regions is that we've surpassed the diffraction limit by about an order of magnitude by doing this. So as another example of what we can image with super resolution, I'll turn to the other structure that we are visualizing here, the chromatin. Now, in this image, we're not going to be visualizing the chromatin. We're going to be imaging a protein that's actually on the surface of the nucleus. The protein here is, in this case, is lamin B1. So over here on the right is our super resolved image of this protein lamin B1, which is essentially showing us the outer envelope of the nucleus. And so here I'll play this video, and what you realize it's not a continuous image. It's actually many tiny points, and each one of these points is a location of an individual lamin B1 that was fluorescently tagged. So clearly, fluorescence microscopy is a very powerful approach for observing subcellular organization, as is cryogenic electron tomography. But one of the limitations of super resolution fluorescence microscopy is the only thing now that I see is the structure that I fluorescently labeled, right? So here we have no context for what's around this nucleus. We don't know anything other than the location of lamin B1. So the goal of my work in combining these two different microscopy methods, cryogenic electron tomography and super resolution fluorescence, is really to keep the best of both worlds, to retain the molecular scale resolution and detailed cellular context provided by electron tomography, and pair it with the single molecule sensitivity and specificity of fluorescence. So the next experiment I'll show is going to follow this workflow here, where I take a sample that's been fluorescently labeled. I'm going to prepare it cryogenically by plunge freezing, that method we talked about. I'm going to load it into the fluorescence microscope and perform cryogenic fluorescence microscopy. I'm going to take that same sample out of my fluorescence microscope, keeping it cold. I'm going to load it into the electron microscope, find the exact same cells that I imaged in fluorescence, and perform tomography on them. And then I have to align my fluorescence information with my electron tomography information. So as a demonstration of what we can do with this correlative microscopy approach, I want to return to this organism, Caulobacter crescentus. So this organism has showed up a few times in this talk. It's a freshwater bacterium, grows in the bottom of lakes and ponds. But the reason we're interested in it is because it asymmetrically divides. So normally when you think about bacteria and they divide, they grow into two identical daughter cells. But Caulobacter is different. When Caulobacter divides, it divides into two very distinct daughter cell types. This mobile swarmer cell type, as I'll call it, and this fixed stalked cell type. So this fixed stalked cell stays on the bottom of the lakes or the ponds where this organism is growing. But this swarmer cell is sort of free to explore greener pastures. So it swims off and it looks for an area that it thinks is rich in nutrients. And it's going to differentiate. It's going to become a stalked cell. And then it's going to begin replication again. So why do we care about asymmetric division in bacteria? Well, if you think about your own human development, we care a lot about asymmetric division, right? You and I, we all start off as a single embryonic cell. And that embryonic cell has to asymmetrically divide many times to differentiate into the different tissues that make up our bodies. So asymmetric division is an essential part of human development. And when it goes wrong, there's real implications in human health. So we're doing what scientists often do. We're studying a simpler system that exhibits some of the same properties to try and understand this process on a fundamental level. So one protein that we know is important in this asymmetric division process is a protein called polar organizing protein Z, or POPZ for short. So biologists love to give proteins very long names, and it can become very confusing very quickly. So throughout the talk, I'll just refer to this as POPZ for short. So POPZ goes to the ends of this bacterium, the poles of this bacterium, hence the name. And you can see that upon division, it leads to an inherent asymmetry. There's this protein at one end of the cell, but not at the other. So we want to take a look at this asymmetry in more detail. So we're going to apply our correlative approach. And so we're going to label POPZ with this label here, photoactivatable monomeric Kate. Again, another very long name from biology, but this is our fluorescent label here, PAM Kate, and we're going to attach it to our protein of interest, POP-Z. We're going to apply our correlative approach. So we're going to take cells that have this, and we're going to freeze them. We're going to image them in fluorescence, and then we're going to image them in the electron microscope. So that looks something like this. So here is roughly the field of view that we're looking at, this dashed box, sort of in cartoon form, in this fluorescence video that I'm about to show you. So as I play this, what you're seeing are single molecules turning on and turning off at cryogenic temperatures. And each time we see a bright spot appear, that's a molecule that we can identify and localize very precisely. We can then take that same sample, we can load it into the electron microscope, find those same cells and perform tomography, and that looks something like this. So here's a single slice from the reconstruction of this bacterium. We can do that same process of coloring in all the different features that we can identify. So again, that includes things like ribosomes shown in yellow. We have our inner and outer membrane. We have a surface layer of proteins. But as this video progresses and we zoom in to the ends of this bacterium, we again just see this void where there's really nothing that we can identify from tomography alone. So we actually just see this abrupt end to the ribosomes here. And so we refer to this space as the ribosome excluded region, because that's really all we can see from tomography, right? If we zoom in on this data, we can see, for example, the ribosomes here, these very dense structures that I'm showing, or trying to highlight here. And then they stop, and there's fluctuations in the density here, but there's nothing that we can identify and say, that's POP-Z, right? We just see these rough fluctuations in density. But now what we can do is we can take our fluorescence information and overlay it on top of this tomography data. And I'll first show you what this looks like in a diffraction limited sense, something like this. So here our fluorescence, our diffraction limited fluorescence information is this pixelated heat map. And you can immediately see the problem with using diffraction limited fluorescence information with electron tomography, right? This spot here in this pixelated image is roughly the entire width of this bacterium. All it really does is tell us that our protein is somewhere in this half of the cell. So now what I'll do is I'll strip away this diffraction limited information and I'll reveal our single molecule localizations. That looks something like this. So each one of these red circles now is the location of an individual POP-Z molecule that we identified and localized, and the radius of these circles is our uncertainty in that position. So now we know very clearly that this protein of interest, POP-Z, is in that ribosome excluded region. But one of the limitations in this work from 2020 is just the small subset of molecules that we're able to identify and localize. And one of the challenges we face is that we would love to increase our excitation intensity and image more molecules and image them faster, but if we did this, our sample heated up and ended up melting. And so coming back to understand that problem more clearly, we looked at the grids themselves, and the grids themselves were really the problem. So remember that this is some metallic meshwork, and stretched over the top of it is this amorphous carbon layer. And again, think graphite and pencil lead. This is very absorptive. And this absorptive layer was really driving heating in our experiments. And so we realized we could do a lot better. We covered that amorphous carbon layer with a layer of silver. And that silver is highly reflective, and it's also a really good thermal conductor. So any heat that the sample does absorb, it gets dissipated very effectively. And that allows us to take images like this one. So here again is another tomographic slice of one of these bacterium, and I'll zoom in on it once more here. So we have ribosomes all out here, and then we have this ribosome excluded region shown in this green box. And now what we can do is we can decorate that ribosome excluded region, again with our single molecule localizations, but now collected on these silver grids, and that looks something more like this. So we have a much denser set of localizations. And what we realized in these experiments and others is that POP-Z is actually forming a droplet in this side of the bacterium. Just like oil and water will form droplets in each other, this is forming a droplet of POP-Z inside the cell. So another way of visualizing this data is the following video. So our green crosshairs here now are our single molecule localizations, and this gray cloud is actually an entire droplet of this protein POP-Z. So what is POP-Z doing in asymmetric division as a droplet? Well, one thing it's doing is it's recruiting other proteins to this microdomain, as I'm calling it, this sort of well-defined region within the POP-Z. And one of these proteins is a protein called SPAM-X. So this droplet here of POP-Z shown in green is bringing in other proteins. And these proteins bring in other proteins. And now we have many different proteins that are going just to the specific region of this POP-Z droplet. And inside that droplet, they're performing chemical reactions together, they're interacting, and that's leading to chemical gradients throughout the cell. So now we've gone from an asymmetrically localized protein to chemical gradients in the cell that can be used as cues for further development. And when we perturb this system, for example, by overexpressing this protein SPAM-X, then we get really aberrant behavior in the cell division. So here we see cells trying to divide in the wrong place. We see divisions that have been stalled. So we don't understand all the different processes that go into this asymmetric division, but there's many things that we have worked out, and new tools like this correlative super resolution are helping us tease this apart and other complicated biological processes just like this. So with the last few minutes of my talk, I want to turn briefly to the future of these methods. And I recently read a great quote from Yogi Berra, which is, "It's tough to make predictions, especially about the future." And I think that's especially true in this area of microscopy, where there's lots of incredibly brilliant researchers all pushing in unique directions. It's hard to know what's really going to be next. But there are a few things that I'm fairly certain of. And the first is that cryogenic electron tomography and super resolution fluorescence microscopy are going to continue to improve. In cryogenic electron tomography, this improvement takes a few different forms. One thing is going to be better hardware, faster cameras, lower noise cameras, better stages, just better microscopes in general. It's also going to be driven by a lot of developments in software. So there's a lot of information in the tomography data that we really haven't extracted. And so there's more software that's being written to try and do this more efficiently and effective. And also, there's going to be a big push for sample preparation techniques. So as I mentioned earlier on in the talk, one of the limitations of electron tomography is the thickness of our sample. So there's a reason that we looked at bacteria in today's talk, because it's harder to work with thick mammalian cells and even harder to think about working with tissues. So there's methods that scientists can employ to thin these thick samples. And one of the methods on the horizon that's really coming into its own is something called focused ion beam milling. So here what we use is a beam of energetic ions to actually chip away at thick samples. So what you're looking at in this video is a large by electron microscopy standards, block of ice. It's about 200 microns thick and a few millimeters wide. And embedded in this ice are some fluorescently labeled worms. And what we're going to do is we're going to go in and use one of these focused ion beam milling devices to extract a thin section from this thick sample. And that looks something like this. So here we'll see those fluorescently labeled worms, and we're using that fluorescence to guide this milling process. So we can go in, using that fluorescence, we can figure out which section we want to take. And then we come in with that energetic beam of ions, which actually chips away at the surface and cuts it out like this. And now we have to extract that. So we use robotic tweezers to come in and very delicately pull this section out. Remember that this is fractions of the width of a human hair. And actually, this is too thick to image by electron microscopy or tomography. So we have to further thin it. So we bring it into another part of the chamber here. We load it onto another sample holder, and we further thin this down to a thickness that can be imaged with electron microscopy. So this is one thing that's on the horizon. If I turn to super resolution fluorescence, it's even harder to predict the future because there's so many different variants of super resolution fluorescence microscopy. But one commonality for super resolution fluorescence is that we all use fluorescent labels. And so there's going to be continued improvement in labels that are brighter, more stable, and more informative. We're going to, of course, push for faster imaging and also reduced aberrations as we image in thicker samples. And this is really opening up some new areas for fluorescence microscopy, specifically areas in structural biology, actually solving what proteins look like in some sense, using just fluorescence microscopy. So here's a reconstruction of a structure called the nuclear pore. So these are pores that are actually on the surface of the nucleus that allow contents to move in and out. And this has been reconstructed entirely using fluorescence. So different structures on this nuclear pore were labeled, and many images of identical structures were averaged and aligned to produce an image like this one. We're also, of course, pushing for live cell imaging that's compatible with super resolution methods. Here's, in some sense, an update of the video that I showed at the top of the talk on using the white blood cell, hunting the bacteria. This is a different immune cell attacking a different type of cell. This is the target cell shown in blue. And so this is something that we can do today using live cell microscopy. Another thing I'm confident in saying when trying to predict the future is that there's always going to be limitations to electron tomography and to super resolution fluorescence microscopy. And these limitations are going to leave open room for these correlative approaches that bring the strengths of these very different imaging methods together and combine them to get a very clear picture of what's going on inside our cells. So with that, I would like to thank the many people who helped make this possible. Of course, my postdoctoral advisor, W.E. Moerner, very talented graduate students in his group that I've been working with for a long time, Davis Perez and Anina Sartor, Wah Chiu and members of Wah Chiu's team, specifically Corey Hexel and Patrick Mitchell, Lucy Shapiro and Samia Saurabh for their expertise in colobacter biology. And I borrowed data throughout this talk from many people. Here's a few of the key personnel. And I'd like to remind people that at the end of the talk, I have the EM grids, and we'll take a look at those in the hallway if people are interested. And if there's any students in the audience, I also have some light microscopes to give out if you're brave enough to ask a question. And with that, I would like to thank you all for your time and attention, and I'd be happy to take any questions.
Okay, so now we can take questions. Actually, this is a great room. You all have a microphone in front of you. When I recognize you, push the little red button in front of it, and then you can speak, and you can be heard here and also on the video that's being prepared. So who'd like to go first?
Please. All right, testing. Yeah, testing. 1, 2, 3.
Yes.
2 questions. Number one, it was great, by the way, as advancement. But questions that I have is, after you freeze a sample, how do you transfer it without it? That's number one question. And number two, do you sputter it with gold before you put it in electron microscope?
Yeah, both are great questions. So transferring these systems under cryogenic temperatures is definitely a thing. There's a lot of infrastructure that's been built that allow us to transfer these samples and also keep them free of ice contamination, because one of the real challenges we have is humid air condensing on these samples leads to an inability of us to image it later on. So there's in terms of transferring the grid after you freeze it, there's something that I call a button. It has a small slot in it with a screw cap that comes down, and you have to move it very quickly from one bath of liquid nitrogen to the next. In terms of sputtering with gold, none of what I've shown today was coated in any way with metals. That is a process we do with some of the more advanced sample preparation techniques to try and mitigate charging.
Yeah, great talk. Very interesting. I was wondering what your success rate is in terms of you take a sample and you post-process in some way, and how often is it just kind of garbage out that you just can't make a structure out of it or something interesting?
I don't know what you're talking about. That never happens. It depends on what your goal is, right? Some experiments are harder. The bacteria that I image are fairly robust. And I'm also targeting fairly low resolution in many of these experiments. I would say that there's a lot of steps where you fail along the way, from the sample preparation to the data collection to the sometimes you just don't see that there's issues until you get to the post-processing. A hard and fast number I can't give you because it really depends on what sample you're looking at, but some samples can be really painful, I can tell you that much.
Okay, it's very nice talk. And I'm curious in these regions right there, what you circle on the red, and you mentioned these are the POP-Z protein.
Yeah.
And can you quantify how many POP-Z protein in that hole?
Yeah, it's a great question. So in a lot of ways, the single molecule localization method isn't the best way to do that. The way we do that at room temperature, and the same thing can be done at cryogenic temperatures, is you start with a molecule that doesn't blink. It's just always on. And then what you do is you characterize the brightness of this when all the molecules are on at the same time. Then what you have to do is bleach all the way down till you can see the intensity of a single molecule. And so from there, you can work backwards to how many molecules were present before the bleach down. So there are ways using fluorescence microscopy to get an estimate of the copy number of your proteins. But the super resolution method is probably not the best way to do it. There's always going to be many proteins that we don't identify from the fluorescence, but from this variant of super resolution on its own.
Okay, so in practice, you can probably estimate the numbers of the protein as a function of their physiological activity.
Yes. There are limitations, but in general, yes.
So the lady in the front row had a question.
Why Toxoplasma Gondii?
Why Toxoplasma Gondii? Well, the truth is we have a great collaborator in that area, John Boothroyd and Liav Zarko. I've worked with them for a long time. It's also a fascinating system to me because that apical complex, right? There's so much in that very small region, and this is an incredibly dynamic system, right? Normally, having looked at a lot of bacteria, they don't have dynamics like this, where this entire apical complex actually, in the process of getting primed for host cell invasion, moves from inside the cell body to outside the cell body. There's structures that are interacting at key moments in time. There's secretion of proteins. So long story short, it's just a really fun, interesting sample.
Please.
Is the can you go back to the apical structure again, please?
Back to the apical structure?
So when it bores into, we're talking about the one, yes.
Yeah.
You can see the process when it actually starts to bore into the human cell or whatever cell.
So what
Do you see that actually structurally it changes the same, or does it just release enzyme just like sperm entering the egg?
Yeah.
I just wonder.
Yeah. So what we've captured here is not the moment of host cell invasion. What we've done is we stimulated this parasite to think that's about to happen. And this is one of the interactions that we see is that interaction between that vesicle and that rhoptery here, that red and yellow structure. But we haven't captured the actual moment that it's interacted with the host cell membrane and how it's pulling itself in. You're right, actually, that many of these structures here, like the rhoptery, inject their contents into the host cell at various moments during this host cell invasion process. And we think that part of the role of this vesicle and the fusion might be related to how we go through many different membranes. We have to go through the parasite membrane, the host cell membrane, to get our contents inside that cell.
Yeah, you point to some other aspect, which is very interesting. I mean, he's talking about blowing up space so you can understand it, but you also have to slow down time to see all the details of the process. I guess it's not obvious how to do that. Maybe in the future.
Maybe in the
That's
future.
tomorrow.
Yeah.
Okay, there was a question up here. Yes.
The little 3 millimeter grid that you're.
Yeah.
Where do they come from? How do you build those?
Well, so you can imagine making them with a stamp or a die and pressing them out. Right now, we just buy them commercially, but in the old days, they were made by individual labs.
Let's take 2 more questions. We'll start with the fellow in the back.
So you were talking a little about how in the future, hopefully there'd be better software developments. I was wondering, like for this experiment itself, what kind of software are you using? Because it looks like on this slide, it's like a semantic segmentation problem.
Yeah, parts of it are a segmentation problem, right? How do I identify different features and color them in? And there's been a lot of advances made in that front using convolutional neural networks and other methods where you can provide it some amount of training data and then it colors in the rest of the picture for you. That's been very helpful because that used to be a very, very time-consuming process. Other parts of the software are a little bit different. How do you identify individual protein complexes that you might want to extract or average to produce high-resolution structures? That's sort of a different problem that uses slightly different algorithms, not a convolutional neural network. But yeah, that's sort of one problem is that coloring in. How do you do that? And I wouldn't say that's solved, but there's been a lot of progress recently in that area with the advancement of machine vision and machine learning, rather. Yeah.
Anyone with the last? Please.
When you were doing the etching of the larger cells to get them thinner and then picking them up with
Yeah.
the tweezers,
Yeah.
I didn't see carefully how thin it was. Do you remember?
The section that I'm pulling out, or that's on the order of a few microns. Yeah.
Wow. Okay. Thank you.
Yeah. So we have to thin it down to a few hundred nanometers to be able to image it.
And so you do it multiple times?
You take it out and you put it into another place, and then you continue to thin it using the ion beam.
Thank you.
Yeah.
Okay, well, Peter, thank you very much. And thanks to all of you for coming. Tell your friends. If you're not on our mailing list, you can sign up on the SLAC website and we'll send you the announcements. These lectures occur every couple months. We'll have another one at the end of March. That'll be about basically how to tune up our new accelerator using machine learning. So these topics spread all over the expertise of SLAC, and we hope they're all fascinating to you. So again, thank you very much. Peter will be out there in the lobby in just a minute to show you the grids and talk some more about this if you'd like. And see you next month.